room. Many people have planned their summers for years around coming to the Sierra Nevada Field Campus. And this was our way of bringing you back together with us, because we are all missing being there as much as you are. And a few years ago, we got the Friends of the Sierra Nevada Field Campus started to kind of catch up on some of the deferred maintenance on campus and boost our enrollment. And so we really appreciate you coming. It's a good time to do an extra fundraiser, since we're not seeing you in person, which any other summer we would be able to show you all the things that are happening on camp, on the facility. So a couple little housekeeping things. I'm sure most of you have been in a Zoom meeting in the last couple of months. There are sometimes technical glitches, and so bear with us if those happen. If you're having trouble, I would suggest log out and then log back in. Sometimes that's a cure-all. Also, just so you know, there's a if you move your cursor, if you're on a computer, down at the bottom, a panel of options will show up, and we have a little poll of just a couple questions for you if you feel like taking that. And we have a Q&A. So if you have questions for any of the instructors, you could put that in there. And if we have time at the end, we'll go over those. And Kristen Muser, one of our art instructors, is going to be managing that behind the scenes. So, and she'll answer questions that she knows the answers to. So I think, the other thing is one of our presentations is going to require a pen and paper. There'll be a little writing exercise. So if you have a chance to go grab a pen and paper, that would be really great. Now, maybe all of our panelists can come on. I'm going to introduce Greg Budney. He will be running the show today. And so Greg was the audio curator at the Macaulay Library of Natural Sounds at the Cornell Lab of Ornithology for many, many, many years. And starting in 1989, Greg would take over the whole field campus with students from literally all over the world. And they came for Greg's expertise in sound recording, and they come for the exquisite quality of sound in the Yuba Pass and the Lakes Basin and the Sierra Valley areas. So he is going to be introducing all of our panelists, which you're seeing right now. And I also want to do a little addition here. I would like to dedicate the webinar to Jim Steele. And Jim is at home recovering from heart surgery. And as he'll watch the recording later of the webinar, but we want to do a shout out to you, Jim. He was director of the field campus for over 25 years. And Jim's the one who created this congenial atmosphere at the field campus. He introduced the idea of workshops and bringing in artists and naturalists and enhancing the program from the biology department at San Francisco State. So we dedicate this webinar to you, Jim, in gratitude for all you've done and for your vision in starting the Friends. The idea of the Friends hatched a long, long time ago, and we've finally been able to bring it into reality. So I think that's everything. Is that ready to take it away, Greg?
I am. Thanks very much, Mary.
Yeah, you're welcome.
First, I want to thank all of you who are attending this webinar. Despite not coming together at the field campus, all of you are in our hearts and minds this summer. We miss seeing your eager faces, eager and attentive faces, and hope you miss seeing our faces as much as we miss yours. I'd like to just very briefly take a moment and explain Friends to follow up on what Mary has already said. For those of you who are not familiar with Friends of the Sierra Nevada Field Campus, it's a not-for-profit group consisting entirely of volunteers dedicated to supporting the educational and experiential mission of the Sierra Nevada Field Campus. Because Friends is a volunteer group, completely volunteer, 100% of the financial contributions made to Friends go entirely to improving the infrastructure of the field campus. So that's certainly among not-for-profits a fairly unique thing. So today's webinar will have presentations from 5 instructors from the field campus: Brian Perry, Andy Trams, J.R. Blair, Sarah Rabkin, and Tanya Baxter. Each instructor will have about 10 minutes in which to provide a quick overview of their class or workshop and provide a brief learning experience to give you a taste of what attending an SNFC course is like. Following the presentations, we'll have an opportunity to take some questions and update you a little bit about the plans for the future of the field campus here, at least how Friends is assisting in that regard. Again, thank you all for coming. The first presentation is by Dr. Brian Perry. Brian is an associate professor in and chair of the biology department at Cal State East Bay. Brian teaches the fungi course for SF State College of Extended Learning at the field campus. The fungi class has a long history at the field campus and is always the first class of the summer. Welcome, Brian.
Thanks, Greg. All right, let me see if I can share my screen here with everybody. Uh-oh, Mary, you need to give me the ability to share my screen.
Okay, I'm working on that, Brian.
Okay.
I'm sorry. See,
That's
these
okay.
these are the kind of memorable, glitchy things that we. Okay, you're on. You're good to go.
All right. Success. Okay. There we go. Whoops. Good morning, everybody, and welcome to our webinar here. And it's a pleasure to be here today. And I'm going to be telling you about the course that I am lucky enough to teach every year at the field campus. And just to tell you a little bit about myself, I am a graduate of San Francisco State University for both my undergrad and my master's degree. And I first came to the field campus and must have been about 1993. I came up and I took a series of botany and bird courses and absolutely fell in love with the place. So for me, being able to come back as a professor and take over teaching the spring fungi course has really been a dream come true. And as Greg mentioned, we are the first class every year. So that first week of June, typically, we're the first ones up there, typically with another class. And the class has been going on for a very long time. It was founded by Dr. Harry Tiers, who was the original mycologist at San Francisco State University. And Harry started teaching the class in 1982, and it's been taught almost every single year consecutively since then. Harry retired in 1989, and Dr. Dennis Desjardins, who was one of his students, took over the class. And Dennis, of course, also replaced Harry at San Francisco State, and Dennis just retired this spring. So he's in retirement now. And then I came along in about 2017 and took over the class. So I've been lucky enough to come up every year and spend a week at the field camp. It's pretty much as I tell my friends and family, it's my favorite week of the year. I absolutely love being up there and having the opportunity to spend time there and share my passion for mushrooms and fungi with everyone. And this is just a picture from our 2017 class. Those of you that are in the Friends group might see a few familiar faces in there. And also, as you know, at the top, June 2019. So last year was the 35th offering of the course. This year would have been, of course, been the 36th offering. So hopefully next year we can do the 36th offering of the class. So this is a typical scene at the end of the week. As you'll see, everyone's got a big smile on their face. We typically take this picture right before everyone heads home on Friday afternoon. The class runs from Sunday evening all the way till Friday morning when we have our final exam. And then we pretty much say goodbye to everyone and wish everyone well. And it's a nice, beautiful morning there you can see, but it's not always that way. So typically we have beautiful weather up there, but being early June, we never know what we're gonna get. So what I tell the students when I'm emailing them before the class starts and telling them what to bring along, I say expect the unexpected. Every few years we get a big snowstorm in early June, and we might have to spend the day sledding instead of collecting mushrooms. This was back in 2012, probably one of the larger snowstorms we've had. And we got a good foot of snow out of the storm. And I have fond memories of sitting in the director's cabin with a roaring fire while pretty much every single attendee at the camp was packed in there. chatting and sipping wine and having a very good time. Well, it snowed outside. And then Jim Steele was the director in those days, and him and the staff were running around pulling beds out of tents as the tents were collapsing underneath the snow load. And they converted the dining hall into an impromptu hotel so that everyone could pretty much have a dry place to sleep that night. So we never know what we're going to get. We might get snow, we might get sun, we might get rain, it might be cold, it might be warm. So we got to be prepared for everything in that first week of June. And as you'll notice, everyone's still got a big grin on their face. So after this snowstorm, the temperatures did warm up, it's melted off, and we found lots of mushrooms. It was still a successful, wonderful week at the field campus. To give you an idea of what the class is like, we go out pretty much every single day, Monday through Thursday. So after breakfast, I lecture for about 2 hours until all the students are nodding off and falling asleep. And then we gather up all of our gear, we get in cars, and we carpool out to different locations around the field campus. And the goal for the week is to go to as many different habitats as we can. And the reason for that is that we want to find the greatest diversity of fungi possible. And I want the students to have the opportunity to collect in as many different habitats as we can. So we'll go all the way up to the top of Yuba Pass, and we'll collect up there in the high elevation red fir forests. We come down a little bit lower and collect in the mixed fir pine forests. And we also collect in things like disturbed habitats. So looking at this slide here, what you'll see is you see a stump right in the middle. So this is an area where the Forest Service has come through and logged a little bit. And we collect in these areas for a number of reasons. Number one, they're oftentimes easy to get around. So you can see this is a nice clear open forest, makes it easier for the students to find the mushrooms. But also there's a number of fungi that like to grow in disturbed soils. So any place where a fire has come through, or if logs have been dragged after they've been cut down, after trees have been harvested, or even in campgrounds. So those are areas where the soil has been compacted. Oftentimes there's fire pits and things like that. These are all great places to find fungi. So native pristine forest and disturbed habitats we'll collect in. We also spend a lot of time in the high elevation meadows, and a large amount of time collecting around snowbanks. And I'll tell you more about this in just a few moments. I'll show you some photographs of fungi, but it turns out there's a really unique assemblage of fungi that come up in the spring in the Sierra Nevada, as well as in the Rockies and the Cascades and any other mountain range that are associated with that spring snowmelt. And I'll tell you more about that in a few minutes. We do collect out in the meadows. Might not seem like a logical place to find, a lot of mushrooms, but it turns out there's some very interesting, pretty rare species that occur in our high elevation meadows. And we also go out and look for those every year. And we also go in places like seepage areas. Again, might not seem like the best place to go and collect mushrooms, but it turns out there's a real large number of absolutely beautiful, interesting species that occur in some of these seepage areas. This is one that we visit every year. This is right up off of Gold Lakes Road. We call it the M&M Marsh because there's two species that grow there, which both start with M. And some years there's lots of water sort of running through the seepage areas. Other years there's very little water and it's pretty dry in there. But every year there's tons of mosquitoes. So this is a spot we go to where you're almost always guaranteed to get your feet wet and get plenty of mosquito bites. But again, it's worth it because there's some really fantastic fungi that grow there. So a typical day would be getting out in the field around 10 o'clock, collecting until about 2:30. We bring our lunches with us. We'll have an impromptu picnic and everyone can stop and eat their lunch around noon. And then around 2:30 or so, we'll all get back in the cars and we'll head back to campus. Sometimes we'll take a break so folks can shower, change their clothes, take a nap, whatever they might need to do. And then we come back together in the laboratory downstairs in the dining hall. And one of the big goals of this week, what this course is attempting to do, is train folks how to identify fungi. So we're going to teach people about fungal diversity, fungal ecology, but we're also going to teach them and train them how to become proficient at identifying mushrooms. And so I bring up a whole fleet of microscopes every year. We set up a whole lab downstairs. Each student gets their own microscope, their own sets of reagents, all the stuff that you need to do that. And then we have a series of guided exercises that will teach you how to go from taking a mushroom, slicing a piece of tissue off of it, getting it under the microscope, and identifying all the structures that you're seeing. And then we also teach you how to use keys to identify fungi. And over the 30 plus years that we've been teaching this class, we've actually written a key to all the mushroom species that occur in the Yuba Pass area around the field station. So it's a really nice resource to have. By the end of the week, my goal is always to essentially have students that are informed, they understand fungal diversity and fungal ecology, but they're also proficient at identifying mushrooms. And of course, as I mentioned before, we have a final exam on Friday morning. So this is, everyone gets an A in the class. It's not that kind of exam where someone's gonna fail or anything like that. but it's really just a chance for you to test what you've learned during the week. And you'll notice that both of the students in this photograph are still smiling in the middle of an exam, so it's a good time. All right, so finally on to some fungi. So as I mentioned before, we have a whole suite of fungi that are associated with spring snowmelt. So as those snowbanks melt, as the temperatures start to warm up and then we get some spring rains, all that moisture goes into the soil, and that results in a big flush of mushrooms that will fruit. And so here's just a small sampling of a big diversity of mushrooms that we find associated with these snowbanks up in the Sierra Nevada. This is my Mycena navicola, one of my favorites. And as you can see here, this mushroom has actually produced enough, body heat, if you will, to melt its way through the edge of this snowbank. Okay, and then over here we have Donadinia. These are these little tiny, almost black goblets that you see growing along the edges of the snowbanks. We have some jelly fungi like Heterotextus alpinus, and then lots of other guild mushrooms that grow in association. So when we take the students up to these high elevation sites, I pretty much just have them walk all along the edges of snowbanks, just looking for the mushrooms that are coming up in association with them. So we have a whole series of mushrooms that we only find growing within a few feet of these snowbanks. So really interesting ecologically. And of course, there's edible species. Now, people come up and take the class for a number of different reasons. Some people come up because they just want to learn about fungi. Some folks are doing continuing education units. And a lot of people come up simply because they want to learn how to identify mushrooms, especially the ones you can eat. And so we do have a handful of edible species that occur in the spring in the Sierra Nevada. The most common one that people are up there looking for, the most common two, I should say, are Boletus rexvaris, or spring king, or spring porcini, that we get in the Sierra Nevada. And of course, morels. Those are going to be species of the genus Morchella. And we have 2 or 3 species that we commonly get up around the field campus. So Boletus rexvaris is the one that everyone goes up in the Sierra looking for. This year turned out to be a really good bolete year. So I was up in the Sierra a few weeks ago and we were pretty much stumbling over these as we were walking through the woods. There were so many of them. And every year there was 1 or 2 students that really focused their attention on finding edibles. So this is Mickey. He came up and took the class a few years ago, and he went out every day and collected 10 or 15 pounds of porcini. He was very gracious and he shared them with everyone. And every night we pretty much had a wonderful porcini filled meal that the staff cooked for us. So really great. And some years there's lots of edibles and some years not so many, but we always find at least a few to share and have the students taste. We also get a handful of rare species. And for me, these, being a mycologist, these are the really exciting ones. So these are species that, we've been collecting up here for 35 plus years, and we only see these every few years, sometimes only every decade or so. So Cantharosobe gruberi here, the one in the upper left, this is one that we didn't see this until maybe 10 or 15 years ago. We've been collecting the same area during the same week for over 20 years. And then one year this thing pops up. We had no idea what it was. Took us a little while to figure it out. Now we know it from about 4 different collections in the Sierra Nevada, but I've been seeing on social media that this spring it's been popping up all over the place. So again, we typically think of this as being rare, but it might just be a situation where we've been up there at the wrong time and we've simply been missing it. And then we get lots of other rare species. There's just a few that are a little bit more photogenic. This is Hygrophorus getzii. This is one that only grows above 7,000 feet elevation associated with snowmelt. So a really specific habitat. We almost never see this. So when a student brings one of these in, it's really exciting for us as scientists, but it's also really exciting for a student to find something that is considered very rare. It's always a big exciting day for them. And then of course there's new species. So as I mentioned multiple times now, we've been collecting up in this same area for 30, almost 40 years, honestly. And during this course, we have discovered many species that were new to science and we have gone on to describe them. And so this is just a small sampling of a few that I think are interesting. This one up here, this is Strobelaris diminutivus, and as its name suggests, it's very, very small. The cap of this mushroom is probably 1 to 2 millimeters in diameter. And the only place you find this is growing inside of cones that have fallen off of pine trees and that are essentially rotting on the forest floor. So when you're walking through the woods and you find something like a big sugar pine cone, you flip it over and you pull back the scales and you look for little tiny dots, you'll find this mushroom. It's really common, it turns out, but it had just been overlooked for years. And to give you an idea of size, those little dots that you see in the background are pollen grains. So a very small mushroom. This is Lactarius rubriviridis. This is one that was collected, probably 25 or 30 years ago, was found only once and then was never seen again. And then Dr. Desjardins found it again about 6 or 7 years ago. So we know it from 2 collections and he described it as a new species. And then this one down at the bottom here, this is one of my personal favorites because I described it. And if you know your Latin and your Greek, as it's applied to scientific names. Keto means hairs, and Tiersia is in honor of Dr. Harry Tiers. And so this mushroom is a cup fungus. And so this white portion here, this white disc, is where the spores are being made. And then it's got this little fringy hairs all around the outside. It turns out this was actually a new genus as well as a new species. So I decided to name it in honor of Dr. Harry Tiers. So keto means hairs, Tiersia in reference to Dr. Tiers. So really the name even translates into Harry Tiers, okay? And then of course we've got many, many more wonderful species up there. So I really hope that you'll come up and take the class one of these years. I'd love to have you all as students. And before I forget, let me thank these folks for the use of their photos. I wish I could say they were all mine, but indeed I do rely on my friends and colleagues for their beautiful photographs as well. So thanks for listening, everybody, and thanks for joining us this morning.
Brian, thanks very much for a terrific presentation.
My pleasure.
Our next presenter is J.R. Blair. Most of you know J.R. as the director of the field campus and the affable fellow who warns us against inviting bears into our tent to share food. J.R., among his many hats, is a biology instructor at SF State, and each summer offers the Birding 101 class at the field campus. J.R.
All right, thank you, Greg. And I want to thank all of you for joining us at our webinar today. It's very exciting to have this going on, and I want to give a big shout out to everyone in the Friends of the Sierra Nevada Field Campus, especially Mary and Greg and Kristen for helping to put this together. And it's really, really exciting. So as Greg mentioned, I teach Birding 101, beginning birding. There's a few other birding classes. So Dave Shuford teaches a class in birding. Jim Steele usually teaches a class in bird identification by song. And Greg Budney teaches a recording class, which typically has to do with birds. I want to give you a little overview of what we do. So as you can imagine, we spend most of our time out in the field. So after breakfast, we head out, we go looking for birds in various hot spots around the area, such as Sierra Valley and Lake Davis and a few other places. And then we come back in the early afternoon for a nap, very important. And then after dinner, we have a slide presentation on birds. Some days we get up before breakfast and go out, and other days we actually stay after dinner, go out after dinner and check out the birds of the area. But I want to give you a little short introduction to what it takes to go birding. All right, so basically I want to talk about bird identification skills. Now, if you look at these two birds, you might consider these to be two different species. They're very different in color and in posture and so on. It turns out that this is a male and female barn swallow. So one of the things that's going to jump out right away, even though this is the same species, is that they're different colors. And a lot of times coloration can actually help with identification. So for example, here we have an American white pelican on the left and a brown pelican on the right. Very clearly different species, different colors. And that works quite often, but other times you have to look at other field marks. So look at this bird, kind of brownish, a little gray, a little brown, spotting on the chest, and thin bill, little eye ring. And then look at this bird. It's a completely different species. So you have to look really carefully to see this. This is Swainson's thrush on the left and a hermit thrush on the right. You see the Swainson's thrush has a little bit heavier spotting. The eye ring's a little bit bigger. And there's a few other subtle features that you might look for in identifying these two birds. Here's another example of two birds that really have the same almost identical coloration. So white stripe down the back, black, otherwise black on the back with spotting, little red on the head, that's indicating that they're males. But if you look at the bill, you can see that the bills are very different in size. So the bird on the left, that's a hairy woodpecker. The bird on the right, that's a downy woodpecker. They have different bill size relative to each other. Sometimes you can tell just from overall shape, you can differentiate species of birds, or groups of birds in this case. So here we have two hawks. The bird on the left is a member of the genus Buteo, which includes red-tailed hawks and Swainson's hawks and a few other hawks that we have in California. And you see that this bird has very broad, relatively broad wings and a relatively broad tail. The bird on the right is an accipiter in the genus Accipiter. And you can see that the wings are narrower and the tail is longer and narrower than the bird on the left, so the Buteo. Now I gotta say that there is, if you saw these two birds side by side, there would be a really big difference in size. However, since they're flying, there's really no reference point that you can take. So, so using size as a metric, unless you have some kind of reference, is really risky. Here's another example of that. So here are two birds, all dark, all black. They happen to be flying side by side, and you can see that there is indeed a size difference. But if they were separate from each other, you wouldn't be able to see that, use that as an identifying feature. Maybe you know that the bird on the left is a crow and the bird on the right is a raven. But you can see if you look at the bill that the raven's bill is significantly heavier than the crow's bill. And also look at the shape of the tail. So that sort of a diamond shape look to the tail is indicative of ravens, whereas a squared off tail is indicative of crows. Sometimes behavior can help you out. So when we are looking at ducks, there are typically two behavior styles that they have. So the ducks on the left, which are mallards, there's a male and a female mallard there. Those are dabbling ducks. And what they do is they go butts up and they're sticking their heads down into the to the bottom of the water there and feeding on invertebrates and vegetation and so on in that manner. The bird on the right is a merganser, and that bird is doing a dive. And it's going to be swimming down to some depths and feeding in that manner. Merganser happens to be a fish-eating bird, but there are a lot of other diving ducks that are that don't eat fish, but still they're divers. Here's another example of behavior. So you can obviously see that these are owls, large owls. Great horned owl on the left is almost exclusively nocturnal. You very rarely see them during the day, and if so, it's typically perched. Sometimes you see them at dusk, but they're most often active at night. The bird on the right is a short-eared owl, and they can be diurnal, which means that they can be seen during the day, and also frequently at dusk or dawn as well. And how about habitat? So here are two birds that very similar in coloration and shape and behavior and all those sorts of things. However, the bird on the right, which is a marsh wren, is really restricted to marshes. We see these in the Sierra Valley, at Steel Bridge, which is one of the places we go. They're very common there. The bird on the left is a house wren, and very unusual to see them in a marshy habitat. Typically, we'll see them in wooded areas, perhaps along rivers or lakes in the trees, but not along the vegetation on in the margins of the waters of bodies of water, excuse me. And here's another pair of birds that very similar, perhaps easy to mistake each other. You can see there's a little bit of differences in their plumage. These are female quails. The quail on the left is a Gambel's quail. And Gambel's quails are restricted to desert regions in the American Southwest and northern Mexico. The quail on the right is a California quail, which is widespread and in other habitats and generally not found in desert habitats. What about the time of year? Sometimes that can help you out. So for example, the bird on the left is a white-crowned sparrow, juvenile white-crowned sparrow. And up at the field campus, for example, we see white-crowned sparrows quite frequently. The bird on the right is a golden-crowned sparrow, and we don't see them at the field campus, but they are pretty common in the Bay Area in the wintertime. They don't breed in the Bay Area, whereas the white-crowned sparrow does, and we can see those year-round. Here's another example. The bird on the left is a calliope, female calliope hummingbird. The bird on the right is a female rufous hummingbird. Up at the field campus, we get calliope hummingbirds quite regularly. They come to our feeders almost all the time. And we do see rufous hummingbirds, but we only see them during migration in the fall in the Sierra Nevada. So we could make some assumptions about that. Sometimes you get fooled about that, but for the most part, the using the time of year or the behavior or the habitat really is a good way to go. And if you take some time, maybe take Jim Steele's course in bird identification by song, you can learn the differentiation of birds by their songs and calls. This is what's called a sonogram. So it's basically a visual representation of a bird song. I don't know what this is, but I bet Greg Budding does, and can look at this and analyze it and tell me what it is. But a lot of birds make vocalizations, such as the cranes on the left, make a chortling sound, very easy to differentiate from other sounds in the Sierra Valley, for example. The bird on the right is a Wilson snipe, and those birds, whereas even though they do have some vocalizations that they use with their mouth and their larynx and so on, but they also make a sound called a winnowing sound, where they do a deep dive, and as the air is going through their feathers, their tail feathers, it makes a very distinctive sound that's readily identified. But sometimes you just have to say, I don't know. Now, you may look at this picture and say, well, there's pigeons and there's crows. Well, the pigeons, yeah, that's pretty obvious. But the crows, I took this picture in Victoria, British Columbia, and there are American crows that show up there, which is the species of crow that we have in California. But there's also another species of crow that looks just like them, called the northwestern crow. And unless you hear them, you don't know what they are. So good luck, good birding out there. And I really want to thank you very much for participating in our webinar today.
Thanks
Hey,
very much.
JR, could you, excuse me, Greg, JR, can you stick around for a minute?
Sure. Yeah,
Oh, let me turn my camera on. I'm a little speechless. We just got an email from a donor who has offered to match our contribution, the contributions that come in from this webinar and from the appeal that's going out next week, up to $10,000.
What?
Yeah.
Wow, that's fantastic.
I know.
That's
So
really
this
great.
is really
Great news, Mary.
exciting.
Thank you for letting me stick around for that.
Oh yeah. So I don't know if you've noticed, but my screen has the address of where you can donate. I swear to God, we did not plan this. This just came in, so you're getting it fresh, everybody. Thank you so much to the generosity of the people who love the field campus so much. So you can go and donate while you're there, or we'll talk to you a little later about the appeal that's coming. So that was my announcement. Sorry to interrupt, but I felt like it was worth it.
Thanks very much, Mary.
All right.
Feel free to interrupt again. Our next instructor is Tanya Baxter. Tanya is a botanist and ecologist who teaches the flora of the Northern Sierra Nevada for SF State's College of Extended Learning. Her enthusiasm for her subject matter will be readily apparent here in but a moment. Welcome, Tanya.
Great, thank you. Well, thank you everyone for coming. We are really fortunate to have an amazing place like the Sierra Nevada Field Campus in the Sierras. I've had an opportunity to work as far south in the southern Sierras, down in Sequoia Kings Canyon, where I got my start, and as well in Yosemite National Park, getting to do surveys post wildfires, as that's now becoming more of a thing these days. And more recently, I've been spending a lot of time in the Feather River Canyon, which is just a watershed over with some wonderful serpentine yellow pine forests. So it's my pleasure to get an honor to take this lead on this course. I had an opportunity to work with Dr. Bob Patterson in my master's program, who many of you may have known through the years as a wonderful plethora of knowledge, just a walking encyclopedia. And having taken taxonomy courses over the years, I was always struck by his breadth of the topic, because we had so many new plant families that are not just familiar with California, but beyond. And that has actually served me quite well in my work over the years. So one of the things I want to talk about with the flora is that we're going to be getting a chance to see numerous different species, genera, and families. And the question often comes up, how do we start? Where do we begin? When we're introducing new people to the flora, as well as those are maybe a little bit familiar with plants and using how to familiarize themselves by plant family. So because we have just a week, this course typically is very content heavy. I will focus on about 8 plant families, and there are some updates that have been going on, fortunately, through the Jepson workshops and also the herbaria there, that are distinguishing some new plant families that we're not as familiar with maybe a couple of years ago. So there's always new iterations that are going to be coming out of this sort of field, which is very exciting that we get to be in this floristic province where we have such an amazing diversity. So here's just a smattering of some ones that I have come across and in and around the field campus. You may familiarize yourself with them. And the plant families I have listed there are just some examples that we'll hit on. Like I said, I usually focus on about 8. And another thing we'll also be doing is going through a range of habitats. So depending on the weather, as it was mentioned, that sometimes we get snow. This last year, we ended up having a rather late snow melt. So we found ourselves working at a little bit lower elevation, which isn't necessarily a bad thing. I actually found it exciting. And if we're even lucky, we may get down as far down to Goodyear's Bar where there's some serpentine. And I'll show you some specialties that I recently found while up there this June. So as you can see in this photo, some of the range of habitats are riparian, the high Sierras there. That center one is the Sierra Buttes, which is a very popular hike. We'll do a full day hike if the weather is given. And then also we'll get a chance to visit some really amazing meadows. So this meadow in the center there happens to have some specialties that we don't get to see in other places. Maybe if we're further north in, say, Mendocino County, that are some parasitic plants that you may be familiar with. And I'll show you some more of those pictures later on the Darlingtonias. So just to give you an overview of how our course usually runs, we start off pretty early in the morning with lectures, and I will break things down, and I give a number of different formats for people to learn, because as I mentioned, we're going to have a large group of people that are going to have different learning experiences. So I want to address all of those, and it may be that I can create the floral formulas. People that are really interested in the science of it can memorize things in that sort of format. I can present those. I love them personally as a student of botany, and I found them fantastic. And then there's also those that just wanted to see illustrations. Illustrations can convey a lot. Fortunately, I can draw. Maybe all the other artists, instructors can tell me how my drawing is, but I usually leave them on the whiteboards for students to see later. And then we'll also use slides as well. And then we'll have dissecting scopes to go through. So we'll spend the first part of the week with those mornings, and then afternoons are going to be hikes, and then later in the week we're going to be doing some full day hikes, I should say. And the dissecting scopes will always be available for students. And I found that the enthusiasm for this topic tends to lead into the evenings where we do return back down to the lab and find ourselves spending time there. Just to note, there is also going to be next year, a new thing that we're going to have. It's a trees and shrubs. Typically, I've taught the trees and shrubs within the course, but there was such a large demand for it. I'm going to make a 2-day workshop just for that on the weekend if you want to just get a nice overview before diving into a full week of botany. So here's just an example of what some of the things we're going to be looking at. As you can see that on the top right photo, that's what you're going to be doing. You're going to be looking through a hand lens a lot of the time and looking at little tiny flower parts. And you'll get to get familiar with a lot of these terms as we go along, particular to each plant family. We're going to pick some really familiar parts. We're not going to get too far into grasses or say the Asteraceae, which may be for another time, just because those will have a lot more to cover in terms of like learning to know different parts. This Clarkia on the left is a Clarkia moschini. There's one of my favorite rarities, and it can be a little bit challenging because there's numerous Clarkias in the area. So we'll get to learn the beauty of those nuances. And actually, I have some poetry that I was inspired by spending so much time with this particular genus that has been a lot of fun to be able to share with students over the years, because we get our heads full of all these terminologies like the calyx and, lanceolate and, what is a halfway divided down the lemma nerve? We don't know. So this is going to be an exciting new way of using your brain as you go forward. And you can see students right there. This was actually in the evening. They were keying in that last photo there. So here's an example of some of the different types of inflorescent shapes you'll see. As I mentioned, the vocabulary is going to be an emphasis. And then also we're going to have some really good books that we're going to be working with. That last slide showed you a taxonomic key. We're going to be using the Jepson manual, as well as the California Native Plant Society local chapters have created some nice books that are local to the area that we'll be able to use with some photos. But I tend to emphasize more on the Jepson only because I want us to get familiar with all this fun, exciting stuff working through these keys. Some other things to note is that we are going to actually have some speakers that are going to be coming. I noticed a number of students really wanted to understand as this week goes on, how can I apply this information? And especially for a lot of our undergrads that are interested in pursuing careers, I find it really important that we talk about things such as like the context of fire ecology per se to botany. So we will have an evening talk and presentation, and that'll be kind of enriching and making it very alive and exciting as we start to talk about the plants playing a role in all these different elevation changes that we go through, and how do they exist where they are. So that'll be very exciting as we kind of progress later in the week. Well, I actually kind of hit a lot of the points that I wanted to cover on this topic on the flora. Hopefully many of you will come out and join us, and I look forward to having you guys up there and getting to share our appreciation for the plants of the field campus. Okay.
Tanya, thanks so much.
Sure.
Our fourth instructor is Sierra-based artist Andy Trams, some of whose work graces the field campus. You may be familiar with it. She's an experienced, engaging, and motivating instructor. In Andy's presentation, she'll share some of what you will learn in her course, Watercolors in the Wild, Sierra Flora. Andy, I'm looking forward to this.
Thanks, Greg. And thank you, everyone, for coming today. Especially thank you, friends, for creating this venue for us all. I'm hoping you can see me. Oh, good. Yes, you can. I first arrived at field camp in the summer of 2006. Sarah Rabkin thought I might be a good fit. And I have been coming back ever since. And I just love being there. I want to thank Jim Steele and Sarah for welcoming me so warmly into the field campus tribe. So today would have actually been our first day of watercolors in the wild. And I'm hoping, let me know guys if you can't see me, because okay, that seems better for me anyway. This would have been the first day. It's not the same to share this way, but I am thoroughly delighted to be here and share a few of the processes that we would have been doing together at field camp. So let's dive in. I'm going to tip my screen down now and turn some lights on. And see if we can see what I'm doing. So first of all, I'm just going to scroll through a couple of images of some paintings that make use of some of the techniques we're going to talk about. So for me, the big questions always are, how can we evoke the beauty and complexity of wild places without having to show every single dang detail, which we will never have time to do? So another question is it possible to sit down outdoors without feeling totally overwhelmed by how beautiful and intricate everything is out there? And then the last thing I think about in teaching and in my own work is, can painting be a responsive process? Can it be something that's about responding to a place in a moment in time, rather than something we're trying to control and plan ahead? So I love techniques that allow me to answer all those questions. And I also love sharing those things. So let's dive in. I'll save these for a little later. Well, there's one more. All right. Something I really enjoy doing is mixing color. And we almost always start that way in our classes together at camp. But today we're just going to skip that and go straight to painting. And one thing that I find really, really helpful is to work with a big brush. So this is a very large, round watercolor brush. And if you really load it up with paint, look what amazing things you can do just by manipulating the brush. It's really fun to twist it and turn it. And after a while, you might even be making some shapes that look a little bit like lupin leaves. You know how they are thick and then they get very delicate right at the very center. So you can try going from the inside out or from the outside in. So because I have a slight slant going here, you can see how the color is pooling. And that's one of the really wonderful things too about watercolors, is now that we have all this wet paint, we can make it do things and see what it'll do. So I'm taking some darker color and just dropping it in to the color that I just put down, which adds a whole nother aspect to things. It's also really a fun and helpful thing to just mess around with your brush. How many different marks can it make? What about a different brush? This is a variation on that theme. This is a more delicate watercolor round. So if I had finer lupin leaves, you know how many different lupins are up there. That's what we need Tanya for. It would be, I'm hoping I can take both classes teach and take Tanya's one after the other, because all those beautiful visuals, those diagrams, oh my gosh. If you were to have studied all those shapes and forms and then come to art class, you would really have a lot going on right away. So another thing that's really exciting about watercolors is this is dry. That was a similar exercise to earlier. Let's see, let's do this side. And if I paint over this now, what's underneath is just going to stay put. Which gives, this is when the it becomes possible to start to suggest how complex things are, growing forms, forests and flowers and all the shapes out there. There are no shapes, unless you're in the desert perhaps, and you can see a singular plant, or up in the Alpine, you'll see isolated plants, but mostly around field camp, where we do all our painting during this class, everything is merging so beautifully in the forest. And how to start to suggest that, layering works really well for it. Another thing that I find really, really fun is to re-wet things. So I can re-wet the edge that I just put down there. And that hard edge will go away. I can leave some hard edges, and say I put some, just take away what I just did there. Now I have these really soft, like if you were to squint your eyes or looking off in the distance, then I can do that wet into wet thing. That's really fun. To just suggest backgrounds in the softness. So we really only see things crisp and clear where we focus our eyes, and painting can be the same kind of situation where you just decide where you want the focus to be. There doesn't have to be detail everywhere. So here's another thing that is just delightful and fun to do, which is glazing. That's just using some transparent color and going over paint that's nice and dry with a soft brush. A big brush is really helpful. I'm using a yellow there and a little quinacridone gold. And it really can create some interesting, it starts to suggest the glow of sunlight in forests. Or if we were to go dark, or blue, and glaze with a blue, that's going to suggest some shadow as we darken areas. So gradually, things can start to resolve, and we slowly begin to figure out what the heck we're doing. And as a painting starts to gel, then I find it's really helpful to slow way down and take out some smaller tools and perhaps work light over dark. So gouache is simply watercolors that have additives that make them opaque. So that allows us to not have to have a plan ahead of time, but we can add bits of light into the dark, which I find to be a really lovely metaphor for our times. Field camp is all about light in the world. This kind of activity is all about light. And I love the metaphor aspects of art. What do things mean? What's the meaning behind things? So the traditional approach for watercolor is that we would have a plan ahead. We would start with our light colors. We'd move gradually to our darker colors, and then we're done. And we don't have any possibility of going back to find the light. But with gouache, we can. So this is something, these wild currants and little mountain violets, we would be seeing those around field camp these days. most likely, or they might be all done by now. This was from earlier in June. And you can see those little delicate currant flowers. I could never have saved those spaces, but with gouache, I can come and find them and add them in. And I just find that to be really fun. It allows me to be responsive. It allows me to pay attention. I don't have to have a plan. And it can be a more meditative process that connects us deeply to a place, to ourselves, to a moment in time. And then in the end, you wind up sometimes with an object, a painting, that's a record of that experience. And I think it's magic. So I hope you'll be able to get out there and paint in the wild places this summer. And thank you so much for coming. Bye.
Andy, thanks for a lovely presentation. That was really inspiring.
You are welcome.
Sorry. Our final presenter of this session is Sarah Rapkin. Sarah is a writer, editor, and serves as secretary of the Friends of the Sierra Nevada Field Campus Board, where her thoughtful and open thinking is invaluable. Sarah's been teaching at the field campus for over 30 years, and for the past decade, she's been leading an annual high country writing retreat. Welcome, Sarah.
Thank you, Greg. And a big thank you to Mary for all the work she's done to make this webinar happen. And thanks to all of you for joining us this morning. It's such a pleasure to be here with some of my fellow field campus instructors and a whole bunch of people who share our love for the place. As Greg noted, I've been teaching at the field campus since the late 1980s. And as for a lot of you, it's become for me a kind of haven where many tributaries of my life path come together. In my case, my training in biology and science writing, my love of the Sierra and of natural history, and my life work as a writer and editor and teacher of writing. In just a couple minutes, I will invite all of you actually to try a bit of high country writing yourself, right where you are, just to give you a taste of what we do at the retreat. So as Mary invited you to do earlier, if you haven't done so yet, if you could have a paper and pen at the ready, that would be great. But first I want to give you a quick overview of what we do in the writing retreat. It's 5 days long. We start with an introductory meeting on Sunday evening, usually in the director's cabin, and we depart the following Friday afternoon. And the class is open to all levels of writing experience. You can be very new to writing as a practice. The retreat also draws people who have been writing most of their lives and published writers. And it's conducive to all kinds of writing too. People write poetry, prose, fiction and nonfiction, and also more outward looking or persuasive writing, as well as more personal writing. So I offer up a lot of guides, guidelines, prompts, invitations, readings to stimulate people's writing, but all of it is open-ended enough that it can lead to wherever people need to go, what their interests and preoccupations are in the moment. People write about a great variety of topics, and really we allow everybody to take whatever direction they want to. The group is limited to about 12. Partly that's because our meeting places don't allow for big crowds. We spend a lot of our meeting time together on the dining hall deck and occasionally in the director's cabin. And that is also conducive to a level of intimacy and comfort and trust, and the kind of support and camaraderie and guidance and inspiration that can be important when you're a group of people writing together. Everybody is always invited to share what they're working on during the course of our writing sessions by reading aloud to the group, but nobody is ever required to do that. We spend a couple of 2-hour periods together each day in which I offer up invitations and people actually write right there on the spot. In addition, I offer the opportunity for one-on-one meetings with me and or with my husband, Chuck Atkinson, who's a poet and former writing teacher, who usually attends the retreat. And then in addition to those things, we have a lending library that's available in the dining hall. People can borrow books and read them. And sometimes we talk about those books in the group. But maybe the most important component of the class is free time. People have a lot of open time when we're not meeting together, when they can write on their own, or read, or hike, or swim, sleep, hang out by the river, spend time on their own or with other people. And I think some of the most fruitful hours are those when people are off on their own or with each other, just being in the place. And that leads me to the last aspect of the retreat that might be worth addressing, is just the question of why a writing retreat in the mountains? I mean, obviously we could be doing this pretty much anywhere, but as those of you who have done some writing up in the mountains know, and most of you can probably imagine, there is a special quality of inspiration that happens up there. And one of the reasons to do this where we do it at the field campus, I think might be best expressed by a poem that I like to share often at the beginning of the writing retreat by the poet Mary Oliver, called The Old Poets of China, who says, "Wherever I am, the world comes after me. It offers me its busyness. It does not believe that I do not want it. Now I understand why the old poets of China went so far and high into the mountains, then crept into the pale mist." So part of the writing retreat idea is the opportunity for getting away from the distractions and obligations of your daily life, and what opens up when you're in this exalted location and able to connect in solitude to the life of the river, the forest, the wildflowers, the lakes, peaks, and also with the currents of your own life. And paradoxically, I think another reason why doing this at the field campus is so valuable is the opportunity for the communal pleasures of the place, the dining hall, the chance meetings with people. That also is enriching and inspiriting in a way that informs our writing. And invariably, by the end, it's really clear that everybody who participates in this retreat is coming away having made some kind of internal shift and grown some new possibilities as a writer, as a thinker, as a human being in the larger world. So as I said, I want to share a taste of what we do by giving you a chance very briefly to do some writing of your own. And just by way of introduction of that, this is the kind of writing we do in the writing retreat is maybe different from what you might be used to in writing. It's writing not so much to communicate with others, at least not at first, but rather to find your way to what you need to know, what you need to say to yourself, a kind of notes to yourself. Certainly it can become the raw material for more polished writing. But what matters most is the places that you can get to in your mind and in your awareness that might be inaccessible without the process. It's a way to pull up thoughts and connect with your own insights and memories and images. So the way we do this is to write very quickly with no editing and no censoring, not worrying about grammar or spelling or being eloquent, as if you're just writing notes to yourself. And really, you don't need to have complete sentences, certainly no grammatical correctness or punctuation, whatever. And I'm going to give you just 2 minutes to try this on your own, and I'm going to suggest a subject to you. I invite you to bring alive a memory of a place where you love to spend time, and perhaps a particular moment in that place. So if you are a field campus devotee, you might imagine yourself back into a moment at the field campus where the memory is still vivid. But any place that you love and care about that's vivid for you will do. So start off now by imagining yourself in that place. What are the smells, the sounds? What does the air feel like? What do you see around you? And as you're ready, just start jotting down notes as if you're writing a postcard to yourself, to try to get as many of the details as you can. Stay as concrete as you can. And keep on writing, jotting down details. See what you can pull up from memory and see if the process of writing itself helps you open up into reliving that experience. Okay, I'm going to set my timer for 2 minutes and we'll write and then I'll come back. About half a minute left. Keep writing. And that's 2 minutes. So I hope at least some of you are feeling a little bit reluctant to stop, as if you've just begun. And if so, I encourage you to keep on writing after the webinar is over. I hope you enjoyed that. I think it can be surprising how little time you need to sort of take yourself into the vividness of a place, and how writing can be a tool for doing that. There's way more in your memory and your awareness than you have immediate access to. And Andy talked about the value of painting and doing visual art as a way of bringing yourself into a present moment. Writing as well can do that. It can bring you back a memory, but it can also take you into where you are right now. So thank you so much for your willingness to participate in this little experiment, and for coming. And we hope to see you on campus next summer.
Sarah, thanks very much for that. Thanks to all of you who've been listening in. The presenters have just described a few of the field courses that take us into the spectacular locations around the field campus. At the end of each day, when we return from the field to the campus is the place where we synthesize and process and reflect upon what we've seen and done, and share our experiences and finds with others, both in our class and that we happen to be attending, and with other classes. And the campus is a remarkable setting, affords a remarkable setting in which to do this. And this is what the Friends is really committed to, keeping that type of exchange going through its support of the campus facilities. So in that regard, we're excited to tell you about our next funding campaign and the project we hope to support. We're trying to raise $75,000 for remodeling the main building's first floor laboratory and teaching facility down on underneath the stairs, if you will. And we hope to improve that space, which has long been utilized by many classes, through the installation of larger windows that will improve the natural lighting, make it a much more engaging and pleasant space to be in, removing little used cubby storage spaces, improving the electrical service and layout so that microscopes for dissection and looking at specimens can be improved, improving the lighting units for each individual microscope. And for those of you who are familiar with this room, there's a series of supporting structural pillars that run the length of that room. And part of what we'll be doing is looking at the feasibility of removing at least some of those pillars and creating less obstruction of the screen and presenter up front for those of you who are sitting there listening to a presentation. And of course, now we have, thanks to the news that Mary just shared, a great leg up on attaining that goal. So I encourage all of you out there who have participated in workshops and courses at the field campus to help make this significant renovation possible.
I think we have a really good shot at it with this matching contribution with some funds the Friends will have left. And this week, an appeal is going out. We're doing the Friends are doing an extra appeal this year because we don't get to see you in person and show you what's happening on campus and get you all excited about what's coming. So we are doing another appeal, and it's just to know that we're going to extend the lab into a similar look and feel to what we've done in the bathrooms down on that level. It's going to be warm and woody and light and a joy to go in there, especially for those early classes who have to come in from the cold. They're going to have a nice warm lab to come into. So I really encourage everybody to donate. You can donate today at the website that's listed here, or, you know, when your appeal comes in the mail, you can send it in that way. Do we have anything else? I don't think we have any more questions, do we, Kristen?
We have one more question. What about rebuilding the bridge so fire trucks can use it?
Good question. Who would like to answer that?
I can talk about that.
Okay.
So that's really, I keep calling it the elephant in the room. It'd be a major undertaking with many hundreds of thousands of dollars in cost. So I think what we need to do is to find a funding source that can provide us with monies of that scale.
Yeah, I see us as partnering with San Francisco State in a capital campaign because it's going to be part of a multi-year vision. Once we get a bridge built and big trucks can come over onto the other side of the river, we have the possibility of creating a winter dormitory, a new building for the instructors to sleep, more classroom space, that sort of thing. But it all hinges on the bridge. Were there any other questions, Kristen?
Yes, there are. There are two more. What was done to the downstairs bathrooms? Who would like to answer that?
Okay. Well, I'll answer it. I don't know if the person who asked the question has been to the field campus, if they remember the dungeon that was. Both the men's and the women's bathrooms were very dark, very cold. The drains didn't really work. They had the portable kind of standing showers. They now have tiled showers. This year the women's ceiling got put in. There's heat, there's light, there's ventilation. The stalls for the toilet are now this beautiful knotty pine. The windows are replaced. There's new sinks and mirrors. It's just a completely fresh, new, warm space. And the men's is getting tiled right now. So by the next couple of weeks, the men's will be completed. It's been one of the fun things when in the summer, like last summer, we were able to get the men a tour of the women's bathroom, and that was really exciting for us. And they could see what was going to happen in the men's bathroom, which is going on now.
There was temporary envy for sure.
Yes.
So we have a couple of more. So one person has asked Dee, I would be willing to pay for more webinars, particularly if they offered more in-depth lessons. So that's, yeah, she said that she'd like to attend all of them. There we go.
Yeah. Well, we've certainly had a lot of people step up to offer, our instructors from the field campus offering, particularly the artists, because they've had to convert their classes to Zoom now. So I did take a glance at the poll, and 95% of the people answered the poll that said yes, they wanted another webinar. So we'll keep you posted. Anything else, Kristen?
That's all I'm seeing. Oh, something just came in. Let's see here. Okay, real quick. It's not a question, but thank you so much to the leaders. Amazing what can be done, conveyed in just 10 minutes. And thank you for the webinar. I attended the field campus many moons ago. as SFSU Cons Bio MA student. Enjoyed the memories your presentations inspired and look forward to returning someday. I'll contribute what I can for the good cause.
Thank you.
Nice to hear.
Well, I just want to do a few thank yous. To unmute yourself, Sarah.
At one point, I saw a question that appeared and went away asking about, is there a mailing address for donating for people who don't want to do that online?
Yes, if you go to the website that's listed here, it does have a PayPal link, but it has the mailing address on how to send a check in.
There is an, Kira answered that question, and so if people want to look at the Q&A, under answered, hopefully that's how they can see it.
I can give the address right now real quick if you think we should. It's 2036 Nevada City Highway, number 94. That's in Grass Valley, 95945. That's how to mail a check. Okay.
And I'll put that in the chat as well for
Okay.
everyone.
Yeah.
Thank you.
Thank you. There will be a recording of the webinar available later. So I want to thank a few people, of course. Sarah Rabkin and I worked on this together, putting it together. The learning curve was steep, I'll tell you, but we're used to climbing mountains, right, Sarah? Kristen, thank you so much for being a techie in the background for us. And my friend Kira Epstein, who refuses to put herself on camera. We're thanking her for mentoring us through this process. And thank you, friends, for contributing, for supporting the field campus. The improvements on the campus have really been instrumental in increasing our attendance. Until we had this thing called a pandemic, we had registered twice as many students at the field campus this summer. So it's making a huge difference, and we couldn't do it without you. So thank you so much. I hope that you contribute generously, often, and soon. And, and thank you all the instructors for participating today. I'm gonna run the slideshow, and so if any of the participants feel like sticking around, I'll continue with the pictures until you feel like leaving. Thanks for coming to the field campus today.
Thanks, everyone.
Thank you, thank you, thank you. It was
Thank
really
you.
fun to be here.
Thanks, everybody.
All right.
Whoa, and what time is it?
11:21. Great job, Mary.
Thank you, JR.
It's great.
Really worth it.
It was fun.
Good job. You attained the learning curve.
Can I put my face in now, or is that rude?
The work really paid off. It was really well done. Thank you.
Welcome. And
Nice
thanks
to
for your
have you.
offer, Tanya. We're still broadcasting, so thank you for offering to do a class. There's yours truly.
Great.
Hi, Annie.
Annie.
It was wonderful. Thank you.